kalibutan. Imo kining tanan.
Initiating virtual tour.
And we are live!
Actually, this is pre-recorded.
Hello there! You made it! Welcome to the virtual tour of the University of the Philippines, Cebu. I'm Annie, and I am a proud alumna of UP Cebu. And between just you and me, I had the best 4 years here in UP Cebu. I came from the College of Communication, Arts, and Design, where we did not learn only the basics in journalism and other communication components, but they also taught us how to become relevant.
You only have one viewer.
My college is also home to the fabrication laboratory, where people meet and converge to use different equipment where they can fabricate things, stuff for the advancement of industrial and product design. All of this comes from the creativity of the Fine Arts program itself. Hey, Sean, why don't you tell them more about the Fine Arts program?
Fine Arts program itself, founded in 1975, is the pioneer formal school in the fine arts outside the national capital. The college is also the host to nearly half a century-old annual painting competition, Jose Hoya Awards, in honor of the famous national artist Jose Hoya, and from whom UP Cebu's Jose Hoya Gallery was named after.
The campus of the premier university here in the Visayas, we have to be good. No, we have to be better, or even we have to be the best, not only in the humanities, but even in the sciences as well. Isn't that right, Sean?
That's right, Annie.
In 2014, UP Cebu CENVI, or the Central Visayas Center for Environmental Informatics, came up with high-resolution maps using LiDAR technology. The use of this technology by UP Cebu CENVI provided government agencies with accurate data for more precise community flood risk assessment and management. This helped local government units make informed decisions based on data science and, in the process, maximizing their financial resource utilization. And in the case of the Naga Cebu landslide of 2018, UP Cebu CENVI's use of LiDAR technology allowed for one of the fastest search and retrieval operations in a disaster in the Visayas.
Helping the community through the sciences is very fulfilling. But we were not just exposed to that. We were also exposed to the social sciences, from political science to psychology, and all of these concepts helped shape our mindsets to become better citizens. In fact,
the College of Social Sciences articulates and addresses global challenges. The college offers comprehensive and appealing programs for high school, undergraduate, and graduate students. The Master of Education program is a non-thesis graduate program designed for graduates of the Bachelor of Elementary Education or Bachelor of Secondary Education who want to pursue their professional preparation through future studies in areas relevant to teacher education.
And of course, there were also budding business leaders in campus. They were the ones who had the nose for business, but had the big heart to be of service.
Under the School of Management, students are not only taught to be decision makers, but the forerunners in marketing and finance. It also hones those interested in further studies of this field through its Master's in Business Administration program.
Today, I am now a practicing broadcast journalist, an opinion columnist, an entrepreneur, a teacher, a better person, really. All of this because of my training and experience at UP Cebu.
Nurtured to create, inspired to innovate, destined to serve. We at the University of the Philippines Cebu are driven by academic and operational excellence.
And that's it. Thank you so much for joining this virtual tour. I'm so happy you took part. Oh, look, Sean, a lot of people are tuning in.
But the tour is over.
Well, we'll just do it again.
Ready when you are, Annie.
Reinitiate virtual tour.
Lupa ng araw Before we proceed with the webinar proper, we would like for you to take note of the following reminders. This webinar will be recorded. We will be keeping tab of the proceedings for this webinar for documentation purposes. You're free to leave if you feel uncomfortable that this session will be recorded. Please mute your microphone during the talk. We want to make sure that the speaker's voice is clear to everyone. We may opt to remove you if you don't follow this request. Refrain from taking pictures or videos of the talk. If you do so, you will be held accountable of data privacy violations if the copy you created leaks into other internet outlets. We wish to keep the identity of our participants private, so as yours as well. Please avoid using the annotation tool. Once the annotation tool is activated, you might unconsciously swerve your arrow towards the speaker's presentation and draw unnecessary lines that may block the participant's view on some texts and images on the slides. If you have questions while the talk is ongoing, you may put them in the chat box. After the talk, you will have a chance to ask your questions. We wish for you to only use the chat box rather than voice because of our time constraint. Don't forget to evaluate. Your inputs on the webinar will help us improve our next webinars. After you have answered the forms, you will be given a proof of participation. Also take note that the evaluation link will be opened right after the webinar and shall remain open for only 15 minutes. Our webinar is currently live streaming on our YouTube channel. For those watching through the YouTube Live, you will get your proof of participation by staying until the end of the session. We will be announcing the instructions at the live chat, so please stay tuned. Before we proceed with the webinar proper.
Good afternoon, everyone. Good afternoon. It's a very sunny afternoon here in Cebu. Welcome to the celebration of the University of the Philippines in Cebu to the National Science and Technology Week. I'm Annie Perez Gallardo, Annie, and I come from the College of Communication, Arts and Design, jump shipping just for today to the sciences because we are here and it's a holistic part of us to be able to appreciate the sciences and its contribution to the forwarding of our society. Now, the National Science and Technology Week is a celebration that highlights the significant contributions of science and technology to national development. And it has since become a platform for heralding these components in the country. It is held every fourth week of November, thus why we are here today. And other activities apart from webinars include applied research, technology transfer, technology and commercialization, giving of awards, meetings, technical and investment forums, technology demonstrations, and technical tours. Now, the University of the Philippines, Cebu, participates in this celebration and is hosting 2 events. Earlier this morning, we were able to take a peek at the different projects and researches that were funded by the government. And this afternoon, we will take a look at the research conducted by some of our faculty members, which their researches are also part of their PhD studies. And now, without further ado, let us open our program and let me call on our ever-dynamic Chancellor for the University of the Philippines, Cebu, Attorney Lisa D. Coro.
Thank you very much, Annie. Good afternoon. Again, good afternoon once again. We are now in the afternoon session of our first day of the 3-day series of our celebration on the Department of Science and Technology's National Science and Technology Week with the theme Science and Technology, Tugon sa Hamon or Hamon ng Panahon, or Siyensya o Teknolohiya, mao ang tubag sa mga pagsuway sa karong mga lisod na panahon. I am really very proud. In fact, this morning we had a very successful conduct of the presentation of the different research projects here in UP Cebu. And now for this afternoon, we will be able to see some presentations of highlights on the dissertations of our newly graduated PhDs. And I am very happy that these young faculty are now back with us And they are not just here back with us to share their new knowledge created in their PhDs, but likewise the outputs itself of their sciences and the knowledge which they had created have great impact to our economic and social development as a nation. So let's listen, and I'm sure we will all have a lot to learn from their presentations. Good afternoon once again. Back to you, Annie.
Thank you so much, Chancellor Coro. And that's right, we really had a fruitful session this morning, and it gets more exciting as we take a look at the dissertations of our PhD faculty who are now back here in Cebu. Now, our talk this afternoon is entitled Going Up, Usapang Pananaliksik. Pananaliksik meaning in Filipino, research. So this afternoon will be filled with research, but not just the technical research, but also practical applications that it would be useful for the society. Now we have a special guest with us this afternoon. She is the Undersecretary for Research and Development of the Department of Science and Technology. She holds a BS Electrical Engineering and MSEE degrees, both from UP Diliman, and her PhD EE from the University of Michigan in Ann Arbor. Her area of specialization is speech and audio signal processing, time frequency analysis, and synthesis. She is the first woman dean of the UP College of Engineering and has been the recipient of several awards in engineering and education, including the Metrobank Foundation Search for Outstanding Teachers in the Philippines. Ladies and gentlemen, let us give a warm virtual applause to Undersecretary Rowena Cristina Guevara.
Department of Science and Technology Secretary Fortunato T. de la Peña, Attorney Lisa D. Coro, Chancellor of the University of the Philippines, Cebu, members of the DOST family and the UP Cebu family, distinguished guests, ladies and gentlemen, magandang agham po sa ating lahat. I'm quite pleased to be with all of you today as we kick off the nationwide celebration of the National Science and Technology Week, or NSTW 2021, with a regional event led by the University of the Philippines, Cebu. For more than 6 decades, the NSTW has been a platform for our outstanding scientists, researchers, inventors, and innovators to celebrate the role of science and technology in national development. The need for science remains during the pandemic and beyond. This year's theme, Agham at Teknolohiya, Tugon sa Hamon ng Panahon, illustrates the need of the nation to close the gaps in health, nutrition, business, livelihood, transportation, peace and protection through science, technology, and innovation. Since 2020, COVID-19 has challenged government leaders to rethink strategies to maintain safe, healthy, and prosperous communities. It was also a time when other branches of government freely collaborated with DOST in creating new strategies to advance their goals. It is worthy to note that the Philippines, through the DOST, is part of the worldwide network of the United Nations Solidarity Trials for Therapeutics and for Vaccines, wherein scientific data from each member country is given due credit and significance. Noting the need for readiness in terms of availability and supply of vaccines, we are investing in the establishment of the Virology and Vaccine Institute of the Philippines, or VIP, the country's premier research facility encompassing all areas in the study of viruses and viral diseases for humans, animals, and plants. Through this endeavor, we're hopeful that the Philippines will soon be at par with the rest of the world in vaccine development. We're also pushing for the continuation of the Science for Change program through the Science for Change bill. Given the rapid demand for innovation during the pandemic, the bill aims to address disparity in R&D funding in the regions and lack of R&D in the private sector. The Science for Change bill would allow DOST to build on the milestones achieved by the program since 2016, which includes the funding of 38 Niche centers in the regions for R&D spread out in 17 regions, including UP Cebu's Center for Environmental Informatics, or CENV. These NICERs are considered innovation hubs in the region, providing capacity building initiatives throughout the country. Another subprogram, the RD Lead, deployed 42 RD leaders in 16 regions to share their expertise and promote regional development through R&D. For business innovation, the Science for Change subprogram, Collaborative Research and Development to Leverage Philippine Economy, or CRADLE, created fruitful partnerships between private companies and the academe and RDIs. We're also assisting private companies in their R&D efforts through the Business Innovation through Science and Technology, or BIST subprogram. R&D is not a small investment, but it is an investment that provides big returns. Beyond infrastructure and programs, the DOST also invests in human resource. This is the reason why DOST recently launched the S&T Fellows Program, which aims to further capacitate the DOST agencies in fulfilling our vision of providing world-class scientific, technological, and innovative solutions leading to higher productivity and better quality of life. It has always been the goal of R&D to deliver lasting change for the benefit of the people. COVID-19 may have crossed borders and created new health concerns. The same process can be applied by our Filipino science community by breaking barriers, opening channels of communication to share research, data, and expertise to ultimately defeat the pandemic. I'm glad to share this day to celebrate NSTW with you, and I would like to take this opportunity to express my sincerest gratitude to our Filipino scientists, researchers, engineers, chemists, and project leaders. Thank you for making change happen and equipping the country through your innovations in fighting, surviving, and hopefully soon defeating COVID-19. Maraming salamat at muli, magandang agham po sa ating lahat.
Thank you so much, Yusek Guevara. Magandang akham sa ating lahat this afternoon. And she is right that research and development is not an easy investment, but it can really help us a lot. And we should break barriers and open our communication lines in sharing our research with one another to advance our society, especially now that we are in a pandemic. It would be such of a big help. Now this afternoon, let us listen to our speakers who come from the Department of Biology and Ecological Sciences. Our first speaker is a member of this department here in the University of the Philippines, Cebu. She is currently pursuing her doctoral studies at the University of Melbourne in Australia, and she is into ecophysiological studies and ecological niche modeling to understand how abiotic stress and disturbance history such as wildland fires influence natural vegetation. She is also interested in predicting how climate change could impact plant species. Ladies and gentlemen, let us welcome and listen to Professor Geoffrey Cadiz.
Good afternoon, everyone. This presentation contains only a portion of the PhD research project that I conducted under Australian context. Now, forest wildfire is one of the global challenges of our times today, especially in areas with fire-prone landscapes, such as in Australia, or in California, or in Benguet province in our country. With a changing climate, wildfires are predicted to occur more frequently and more intense. And it is no wonder because weather is one of the three factors that influence fire behavior. Dangerous wildfires are often associated with extreme weather condition, that is, a combination of very high temperatures, very strong winds, and a very, very low humidity. Of these three factors, the amount of fuel is the only one that can be directly influenced through human intervention. The fuel for wildland fire is the vegetation. The abundance of fuel or vegetation and whether it is flammable or not strongly impacts the flammability of a vegetation community. If flammable species can get really abundant or super dominant, it can strongly influence the flammability of vegetation community by playing a key role in defining the physical and chemical properties of the fuel bed. Just like the dominance of the eucalyptus trees in Australian forests that make these forests very flammable. Another example is the dominance of gamba grass that invades the savanna in northern Australia. This grass causes higher intensity fires by increasing the fuel load. For invasive alien species like gamba grass, the drivers of their invasion success and dominance have been studied. But there are native species that are behaving like alien invasives, that is, they become unusually abundant within their natural range, giving negative impact to the vegetation community, but studies on why these native species behave as such are limited. So I ask the question, how does a native plant species become prolific within its range? In the context of forest wildfires, this question is crucial, particularly for flammable species. Knowing and understanding the drivers of abundance of these wildland fire fuel are relevant in managing the fuel and in understanding flammability drivers and feedback. In a broader ecological sense, understanding how a native species can become prolific within its own range can help us understand how native species can be released from the environmental constraints of abundance and behave like invasives. The focal species of this study is a flammable scrambling grass that is found in the understory of forests in southeastern Australia. The forest wiregrass increases the flammability of the forest when abundant. Wiregrass can form a continuous cover of live and dead vegetation on the forest floor, and this makes fires to spread faster horizontally. In addition, wiregrass can climb up tree trunks, and so it can facilitate the spread of fire from the forest floor to the tree canopy. Wiregrass has been reported to grow unusually abundant in certain areas. The role that environmental factors play on the tendency of wiregrass to become prolific in certain areas is not well understood. This poor understanding should be addressed because of the flammability risk this species poses when abundant. The objectives of the study include identifying the highly suitable habitat of wiregrass, which was addressed through ecological niche modeling. Second is investigating how the environmental factors influence wiregrass abundance within its range in those highly suitable habitat. This was addressed through modeling the abundance of wiregrass. Lastly, it's investigating the effect of resource availability, specifically light and water, on wiregrass biomass, and this was addressed through experimental manipulation in a glasshouse. Now, since my time is limited, I will not go into the details of the methods and results, but rather go straight to the major findings. But the results of the second and third objectives can be found in these two research articles recently published in international peer-reviewed journals. The study found out that wiregrass biomass is high when the water resource is not limiting. This is consistent with the fact that this species is found unusually abundant in the understory of the wet forests. Also, wiregrass cover is high in forest stands with reduced canopy cover and increased light, and in recently disturbed areas. The results of this study strongly suggest two salient factors that cause wiregrass to become prolific within its range. First is increased resource availability, and the second is disturbance. The results of this study are consistent with other native species becoming unusually abundant within its range. For example, Pivelo and colleagues in 2018 reported that anthropogenic disturbances, that is habitat fragmentation, forest cut formation, and wildfires, are the key drivers of the explosive population growth of Brazilian superdominant native species. Another example is the dominance of the native understory fern, the Canadian haze-scented fern, which has become invasive in its own range due to increased understory light availability following reduction of canopy cover and the removal of competitor plant species by herbivory. Now, interestingly, these two conditions correspond to two hypotheses explaining the success of exotic invasive species in their invaded community. These are the increased resource availability hypothesis and the disturbance hypothesis. The increased resource availability hypothesis proposes that the success of an exotic species in invading a new site increases with the availability of resources, while according to the disturbance hypothesis, an exotic species has a higher success invading a disturbed site than a relatively undisturbed one. So it appears that both the native wiregrass and exotic species exploit the same opportunities to become abundant or dominant. On a final note, what is the implication of the findings of this study on forest flammability? The results suggest that the wiregrass has the potential to create a positive fire flammability feedback in the productive forests in Australia, akin to the grass fire cycle in other systems. When wiregrass is abundant, fires are more likely to ignite and spread. The smoke from fire stimulates the seeds of wiregrass to germinate after fire. In addition, the fire disturbance could significantly reduce the canopy cover in the forest and increase the resources. These conditions can promote wiregrass dominance, which may in turn increase forest flammability. Studies such as this that seek to understand the dynamics of vegetation abundance can help us understand the dynamics of flammability drivers and feedback in fire-prone systems. Before I end, I would like to acknowledge and show my big appreciation to my thesis supervisors, Jane, Tom, Trent, and Alan, and to the members of my advisory committee, Craig, Claire, Rebecca, and Matt, and to the rest of the individuals listed here who belong to the support staff of the University of Melbourne, the fieldwork team, the Glasshouse experiment team, and to all those who have extended support for this research project. And a very big thank you to the following funding entities: the Victorian Government's Department of Environment, Land, Water and Planning in Australia, the Melbourne International Research Scholarship in Australia, the RHD Student Research Accelerator Fund, SEFS, of the University of Melbourne, Australia, and the PhD Fellowship Program of the University of the Philippines System. Thank you very much.
Thank you also, Professor Geoffrey Cadiz. Now, I also want to remind our participants who are here in the Zoom room that should you have any questions to any of the researchers presented, please type them on the chat box, and we have time allocated this afternoon for you to ask them, and the selected speaker will answer your questions. All right. Let's go to the reaction to Professor Cadiz's research. He is a Professor Emeritus from the Institute of Biological Sciences of UP Los Baños. She was conferred by NASP with the rank and title of Academician for her significant contributions as a scientist, educator, and mentor in the field of mycology and environmental biology. Her areas of specialization are plant ecology and environmental biology. Let us welcome Dr. Virginia Cuevas as she gives her comments and reactions and insights to Professor Cadiz's research. Good afternoon, Dr. Cuevas.
Good afternoon.
I'd like to thank the organizers of this symposium for inviting me to react to the paper of Professor Geoffrey Cadiz. The paper presented by Professor Geoffrey Cadiz, titled Forest Wildfires, is about the ecology of forest wiregrass, Tetrasea juncea, a grass native to Australian forests. It grows naturally as an undergrowth species in the forests of northern Australia. Through a well-designed research methodology and correct framework, Professor Cadiz was able to analyze the factors that enable this wiregrass species to overcome environmental constraints and become very prolific and behave as an invasive species. Growth of the grass is favored by high moisture condition and by disturbance of its forest habitat. Regular disturbance in the forest, such as forest fire, gives the grass to exploit since there is abundance of sunlight and more space for colonization. Moisture availability is provided by the prevailing climatic condition of a forest habitat, and repeated fires create a disturbance. With these favorable conditions, huge biomass of the grass accumulates, which serve as good fuel source of forest fires. Fire and biomass accumulation reinforce each other and become a positive feedback mechanism to perpetuate the grass dominance and proliferation. Information and knowledge gained can now be used for management of forest fires. I congratulate Professor Cadiz for her excellent research. Taking cue from the theme of the National Science and Technology Week, Agham at Teknolohiya, Tugon sa Hamon ng Panahon, I expect Professor Cadiz to apply her knowledge of fire ecology in addressing an important issue in our country, forest fire management. Wildfires frequently occur in our country and destroy both natural and man-made forests. This is most crucial today, especially during El Niño events, where drought instigates the occurrence of fire. In other countries where fire is a natural selective force, in forest ecosystems, such as in Southwest Australia or in Mexico, fire stimulates biodiversity. However, fire in the humid tropics, like our country, fire has resulted in the loss of biodiversity due to forest habitat destruction. Fire also paved the way for invasion of forest lands by kugon and other grasses. It leaves the soil bare for some time, which is then subjected to soil erosion during rainy season. Repeated fires led to severe soil degradation. We now have in our country around 3 million hectares of grassland areas with low productivity, which were formerly forest lands. Our government has spent millions of pesos for reforestation only to go up in smoke. Fire management, therefore, is an important ecological necessity, especially in watershed areas. For water security of our country, watersheds should be kept forested. Fire should be prevented. And I believe that in this area of fire management, Professor Cadiz can give large amount of inputs. Thank you for inviting me. Good afternoon.
Thank you also, Dr. Virginia Cuevas, for that reaction on Professor Cadiz's research. And hopefully, the research could extend to the Philippine context soon, as fire ecology is also important. We may not have a lot of forests like in Australia, but we agree that fire should also be presented. Now, let's move on to our next speaker, to our next research. He is an assistant professor in the Department of Biology and Environmental Science, the College of Science in UP Cebu. He obtained his PhD in environmental biology from the University of Essex, United Kingdom. His research interests include, but not limited to, the following areas: microalgal physiology and ecology, climate change ecology, environmental biology, and thermal biology. He is currently the project leader of Project SHIPS that aims to assess and predict marine heat waves and their impacts in the Philippine seas. Now let's have the presentation of Dr. Edulantes.
Good day, everyone. I am Brisniv Edulantes. I am here to talk about my PhD research work that I conducted at the University of Essex. The topic is about the thermal responses of marine phytoplankton and its implication to their biogeography in present and future oceans. These are phytoplankton. Phytoplanktons are unicellular photosynthetic microorganisms that are drifting with the current in the euphotic layer of the oceans. They are ecologically important as primary producers and regulators of biological carbon pump. However, under certain circumstances, there are certain species that produce harmful algal blooms. These species are able to produce high biomass that leads to hypoxia and subsequently leads to fish kills. Others are able to produce toxins that accumulate in fishes and shellfishes, and if consumed, can cause poisoning and may be lethal in humans. Here is a global distribution of HAB events from 1977 up to the present, showing that HAB events appear to increase globally. These HAB events can be caused by toxin-producing cyanobacteria, diatoms, and dinoflagellates that can cause shellfish poisoning. This global expansion can be caused by increased monitoring effort, cultural eutrophication, transport of HABs, translocation of shellfish stocks, and climate change. Excessive emissions of carbon dioxide in the atmosphere lead to both ocean warming and acidification, which may provide favorable conditions for harmful blooms. Their duration, intensity, frequency, and toxicity may increase in response to changes in the climate. Hence, it becomes very crucial to be able to assess the sensitivity of non-toxic and toxic phytoplankton to changes in the temperature, which is projected to change with climate. The main goal of this research project is to investigate how temperature limits biogeography and ecophysiology in marine phytoplankton. To achieve this goal, this work conducted the following independent studies. The first study investigates the thermal limits to the current biogeographic distribution of marine phytoplankton by analyzing the sea surface temperature of the species occurrence. The second study examines the biogeographic and phylogenetic pattern of thermal biology in marine phytoplankton using physiology and occurrence-based thermal traits. The third study conducts the growth experiments to test the temperature dependence of growth and toxin production in marine phytoplankton. The fourth study conducts competition experiments to examine the effect of warming on growth and competition in marine phytoplankton using marine dinoflagellates as test organisms. The last study predicts the biogeographical responses of non-toxic and toxic phytoplankton in present and future climate scenarios using mechanistic and correlative niche modeling. The last study makes use of the species occurrence data and physiology data collected in the previous studies. At this juncture, I will provide an overview of the approach used in each study, and I will summarize its key findings. The findings should be interpreted with caution in consideration of caveats with the approach. In the first study, an analysis of the global dataset of sea surface temperature of the species occurrence was conducted to examine the global patterns in the realized thermal niche and geographical range of marine phytoplankton. Overall, The findings shed light on the complexity of biogeographical patterns of marine phytoplankton species, which do not necessarily conform to classical macroecological rules, and this complexity is partly explained by climate variability, habitat availability, and/or diversity. In summary, the following patterns have emerged from our analysis. First, the non-monotonous latitudinal pattern in the niche breath is consequent of symmetry between the thermal limits. Second, the narrowing niches in the tropics is due to the reduced seasonal variation in the climate, consistent to Johnson's rule. Third, the latitudinal pattern in geographical range size of the marine phytoplankton invalidates Rappaport's rule, but it is explained by diversity and habitat availability. Fourth, the direct relationship between the niche breadth and range size links diversity effect to the latitudinal trend in thermal niche. From these observed patterns, we conclude that the species in tropical oceans have a narrower range, making them more vulnerable to ocean warming than those in temperate oceans. In the second study, the global patterns of physiology and occurrence-based thermal traits, thermal sensitivity and warming exposure, and vulnerability in marine phytoplankton were examined. Generally, the findings indicate that the variation of these thermal attributes can be attributed to the biogeography and phylogeny of marine phytoplankton. Key findings in this study are summarized as follows. Physiology and occurrence-based thermal traits are congruent but not equal. Second, the inequality between these traits and the thermal sensitivity may vary across latitude, thermal affinity, and thermal specialization. Third, exposure and vulnerability to warming vary non-monotonously with latitude. Fourth, interspecific variation in the thermal attributes is evident. Fifth, phylogenetic signals are present, but no clear evidence of the presence of phylogenetic conservatism in the thermal attributes. The study has identified emerging patterns of thermal attributes in marine phytoplankton, contributing to our understanding of how this species responds to climate change. In the third study, plate-based and tube-based growth experiments were conducted to determine the growth response of non-toxic and potentially toxic strains of phytoplankton. Generally, the results of the study reveal a symmetrical pattern of the thermal growth curve in these model organisms, suggesting that their growth is more sensitive to warming than cooling. The data obtained from this present study was supplemented with the datasets compiled from laboratory culture experiments to allow comparison with an adequate number of observations. The results of the analysis of the pooled datasets show that the maximum growth rates and the thermal traits are comparable. Furthermore, the findings reveal a clear trade-off between the maximum growth rates and the thermal traits in marine phytoplankton, but show evident trait-environment relationships. The results also demonstrate that nearly all the non-toxic, potentially toxic phytoplankton were thriving within the thermal safety zone in the present climate scenarios. However, the warming vulnerability is comparable between non-toxic and potentially toxic phytoplankton. Another objective in the third study is to examine temperature dependence of toxin production. Here, tube-based growth experiments were conducted to examine the dependence of toxin production to temperature, using toxigenic strain of Prorocentrum lima as test organism. After the growth experiments, algal samples were collected, analyzed their toxin using UHPLC MSMS system. The strain has variable concentrations of acetic acid and the dinophysus toxins across all experimental conditions. The strain also has cellular contents of toxins that are comparable to the reported values in the literature. Furthermore, the concentration, cellular content, relative composition, and cellular production rate of toxins in this strain was temperature dependent. The findings also present an inverse linear relationship between toxin production rates and growth rates in this strain. Overall, the results in this study improve our current understanding on the toxin production in marine dinoflagellates, which have a potential implication on the toxic blooms in future climate scenarios. In the fourth study, competition experiments were conducted to examine the effect of increased temperature on growth and competition in marine phytoplankton using dinoflagellate as test organisms. Overall, the findings provide new insight on how warming influences interspecific competition in marine phytoplankton. The key results of this study are as follows. Temperature limits growth and competition. Second, growth response to temperature depends on the interacting focal and competitor species. Third, interspecific competition modifies temperature dependence of growth. Fourth, warming alters interspecific competition. Fifth, growth rates strongly influence competition. And six, ecological response to warming is predictable. This study helps the advancement of our current knowledge on how species respond to climate change in the presence of competition. In the last study, correlative and mechanistic ecological niche modeling were applied to examine the effect of climate change on non-toxic and toxic phytoplankton species. In the correlative ecological niche modeling, the species occurrence data collected in the first two studies were correlated to environmental data to forecast the suitability of a particular habitat for the species. In the mechanistic ecological niche modeling, the thermal growth curves obtained in the third study can be used to establish a causal relationship between species distribution and temperature. Both the correlative and mechanistic ecological niche models were used to determine the shifts in the latitudinal limits and range and changes in the habitat stability, diversity, community composition, and relative proportion of non-toxic and toxic phytoplankton. As shown, most of the species are expected to experience no change or poleward shift in the lowest and highest latitude at which they can exist. It is also expected that the species range may expand, contract, or remain unchanged in the future climate scenarios. It is predicted that the percentage of a new and lost habitats in the future may vary across phytoplankton species and between non-toxic and toxic species. It is predicted that the climate change will decrease the diversity in lower latitudes and increase diversity in higher latitudes. It is predicted that more changes in phytoplankton community composition is expected in tropics as compared to the temperate region in the response to climate change. As per mechanistic ecological niche model, it is expected that the relative composition of toxic species decrease in lower latitude. However, this projection is different from the correlative ecological niche model that show a complex latitudinal pattern in relative proportion of non-toxic and toxic phytoplankton. In this research project, I have provided new information about the thermal responses of marine phytoplankton. The main findings of this thesis are as follows. First, the current distribution of marine phytoplankton is limited by temperature. Second, their thermal traits are contingent on their biogeography and phylogeny. Third, their growth and toxin production is affected by temperature. Fourth, interspecific competition in dinoflagellates is altered by increasing temperature. Fifth, latitudinal range, habitat suitability, diversity, and relative composition of non-toxic and toxic phytoplankton species are expected to be altered in future climate scenarios. The findings of this research advance our current predicted understanding on the ecological responses of marine phytoplankton to climate change. In particular, the information collected in this project can be used to develop models to predict climate-induced ecological trends. Accurate predictions are challenging to produce, but existing models are useful to improve our insights of climate change biology of marine phytoplankton. I would like to use the opportunity to thank the following people and institutions who have contributed to the success of this research work. Thank you very much.
Thank you also, Dr. Brisnev Edulantes. He was also with us this morning when he talked about Project Ships. We have invited another guest to react on Dr. Edulantes' research. She is a biological oceanographer who obtained her PhD in marine biology and fisheries at the Rosenthal School of Marine and Atmospheric Science at the University of Miami in Florida, USA, on a Fulbright Scholarship and a Maytag Fellowship. Right after her PhD, she came back to the Philippines and joined the UP Marine Science Institute, where she is currently an associate professor. Her research is centered around understanding the dynamics of the base of the food web of the ecosystems, especially the primary producers. And she uses an arsenal of field, laboratory, and computational modeling approaches. She is currently heading efforts to provide a decision support system for HABs in the country. Let us welcome Dr. Aleta Iniguez to react on Dr. Edulantes' research. Good afternoon, Dr. Iniguez.
Good afternoon, everyone. Thank you very much for the invitation to react to Dr. Edulantes' presentation. Great presentation and study. I'll just say that actually I've met Brisnev a few times, but not in the Philippines. And I'm really happy that he's back here in the Philippines after his PhD. And from my reaction, I think I'm happy to hear someone working on phytoplankton, because I think that's really rare still here in the Philippines on the especially in this particular kind of area where it's a lot of data analysis and modeling as well. So what I'll do is here in my reaction is kind of emphasize some of the aspects that he discussed and frame it in a how this can potentially be applied here in the Philippines and the exciting things that we can do going forward. So I will just share my screen. Okay. So as Dr. Adulantes has already discussed, we have the phytoplankton, which are very tiny, but they're quite large in terms of their function locally and globally. And they're crucial parts of the carbon or the carbon cycle as part of the biological pump in the ocean, whether it's a carbon sink or as a carbon source. And of course, they are very much key in terms of the productivity of our fisheries, since they are situated at the base of the food web. And without them essentially supporting that food web, then that fisheries will collapse. But as Dr. Adulantes showed, there are also the types that are not so good. So they're tiny, but can also wreak a lot of havoc, large havoc, in terms of harmful algal blooms, which happens when particular species grow or accumulate in certain numbers, and they're harmful if there's toxin or there's depletion of oxygen due to their bloom, which leads to fish or other marine organisms mortality. And I think one of the aspects that Dr. Adulantes showed, the responses can differ. So the type, the number, and the sizes of the plankton in the community affects the structure of the food web and the cycling of carbon and other materials through the food web. And different physical and chemical factors impact those types, numbers, and sizes of plankton. So temperature, as Dr. Adulantes showed, is one critical factor, physical factor. And I think what would be quite interesting is to look more into the interaction of temperature with the other factors, especially here in the tropical system, where there's variability in those other physical and chemical factors. So I'd like to emphasize as well that, of course, the Philippines is an archipelagic nation that is highly dependent on our seas in many different ways. Our 62% of our sizable population living in coastal areas, and the contribution of the ocean economy, particularly our fisheries and coastal tourism and so on, providing about 7% of our GDP and employing a significant proportion of our population. So in that fisheries, the phytoplankton, as I said, is key. So here in the Philippines, we don't have as much of a handle in terms of their responses, their ecophysiological responses, including thermal responses, compared to the temperate areas, which Dr. Adulantes has also looked into more. So there's a lot of literature in terms of the species on phytoplankton species in temperate and in higher latitudes, but we don't have as much information here in the Philippines. But for example, if you're eating sardines, especially the bottled sardines, those come from the Zamboanga del Norte Peninsula, and the phytoplankton are a crucial aspect of their diet in that system. And interestingly, even in that small scale, not even in that global scale that Dr. Adulantes was referring to, there are differences in their distribution. So for example, here in the Dipolog Bay and the Sindangan Bay areas, there were distinct differences in the types of phytoplankton that we found and their density. And but that's considered one upwelling area. So are these patterns consistent through the years? What's causing these patterns? We don't know. Is it just the temperature difference? Is it upwelling, other physical factors, or a combination of these? We don't have a good handle on that right now because we don't have a consistent monitoring information on this. We've on the other part of the Philippines, on the Pacific seaboard side, which is also a critical fisheries area, we've seen shifts in productivity due to changes in ocean circulation, which is affected by temperature changes. So we found that between years, because of the change in how the water circulates, there's different water masses that influence the area, which changed the chlorophyll, drastically decreasing the chlorophyll in 2012 versus 2011, which lowered the abundance and also changed the species composition. So are these, how are these related to temperature and the other factors as well? And of course, in terms of the HABs, we have a lot of issues with harmful algal blooms around the country. The main species is Pyridinium, but there are other species that we've been observing, though less so. But as Dr. Adulantis said, there can be a spread of, in other countries or in globally, there can be an impact in terms of the temperature and other factors affecting the distribution of these toxic species. So here in the Philippines, we don't know yet how exactly temperature and the other factors can influence the distribution of these HAB species that we already have here in our embayments. So this is my last slide, which is a lot full of questions. I think as I said, I'm very thankful that Dr. Adulantis is back, and maybe we can get more into the answering a lot of these questions. So some of the basic aspects is first understanding the distribution of phytoplankton around our waters and their basic biology, just as he did in terms of the ecophysiology and with relative to temperature, and it would be good to extend that to other factors. Their basic seasonal and interannual differences, what their roles are in the ecosystem functions and services, and of course, the impacts of climate change on how they're spatially and temporally distributed, and ultimately the ecosystem functions and services that we rely on. Thank you.
Thank you so much, Dr. Iniguez. That was a very comprehensive reaction. And just like her last slide, maybe some of you here in the Zoom meet also have questions, queries, and point of clarifications for the research presented. Please type them in our chat box, as we have allocated a few minutes after all the presentations for the researchers to answer these questions. Thank you so much again to Dr. Aleta Iniguez for that reaction. Now we move on. This person still is part of the faculty of UP Cebu, and she finished her Bachelor of Science in Chemistry at the University of the Philippines, Visayas, Miagao, Iloilo. With her continued pursuit for learning, she obtained 2 master's degrees. 1, Master of Chemistry, 2, Master of Science in Environmental Studies. She earned her PhD in Chemistry at the University of Aberdeen, Scotland, United Kingdom. Her PhD work was mainly focused on natural products discovery and biosynthesis. In her research, she was able to isolate novel and new compounds with excellent antibacterial bioactivities from Streptomyces bacteria. She also hopes to discover more bioactive natural products with potential for further drug development using local samples. And she was also with us this morning discussing her BSF project. Let us welcome this afternoon Dr. Flordelis Maglani.
As the world continues to grapple in combating and controlling COVID-19, the threat of antimicrobial resistance continues to rise relentlessly, rendering the current antibiotics ineffective for common infections. Antimicrobial resistance is a silent pandemic that cripples and threatens the very core of modern medicine. Since the emergence of the COVID-19 outbreak, there has been increased global consumption of antibiotics. It has been reported that 72% of hospitalized COVID-19 patients received antibiotics even when it is not medically prescribed in most cases, worsening the antibiotic crisis. This figure illustrates the antibiotic pipeline from 1930s up to the present. Those classes of antibiotics that were obtained from chemical synthesis and those that were obtained from microbial sources marked in green. We can see clearly in this figure that most antibiotics were discovered from microbes. Among the microorganisms, Streptomyces has been the most prolific producer of antibiotics. It has produced over 2/3 of the naturally derived antibiotics in current clinical use. The period between 1940s to 1960s led to the discovery of many useful drugs, and this period was dubbed as the golden age of antibiotic discovery. However, there was a substantial decline in the late 1980s to the present, wherein a very limited number of new scaffolds or only derivatives from old classes of known antibiotics are being isolated. And this crisis is compounded by the emergence and spread of multiple drug-resistant pathogens. The alarming antibiotic crisis and the dwindling clinical pipeline urgently call for the discovery and development of new antibiotics. And so today, I will give you a glimpse of my PhD research and my journey to discovering new and novel bioactive molecules. The title of my presentation is Streptomyces sp M837 Natural Products, Discovery, Biosynthesis, and Biological Activity. Due to limited time constraint, I will only highlight the most important aspects of my PhD work. Streptomyces sp M837 was isolated from the rhizosphere of a Morashar bark cloth tree or Antiaris toxicaria, growing in the University of Ghana Botanical Gardens, Legon, Accra, Ghana, Africa. I cultured the M837 strain and nutrient medium, and from this strain, I was able to isolate many different classes of compounds, and these are just a few of them, including siderophores, polyketides, and carbazoles. We have named these legonoxamine B and acramycin A in association with Legon, Ghana, Africa, where the samples were collected. Moreover, I also studied the biosynthetic pathways of these compounds. For instance, the production of acramycin A is poorly expressed in the wild-type strain due to the presence of the repressor ACCJ gene. Bioinformatics analysis showed that ACCJ gene encodes for multiple antibiotic resistance regulator, and we speculate that this gene is a negative regulatory gene that suppresses the production of acramycins. Hence, I carried out the deletion of this ACCJ gene, and lo and behold, after deletion of the gene, I was able to isolate many different acramycin congeners from the mutant strain, acramycin A to K, including known compounds naphtacimycin B1 and phasamycin C. And these acramycins are installed with chlorines at various positions in the acramycin structure. Some of them are dichlorinated, trichlorinated, and tetrachlorinated. Comparing the fermentation titers of the wild type and the mutant, wild type where their genes are still intact, and the mutant where the ACCJ gene has been inactivated, the wild type produced acramycin A in 0.2 milligrams per liter, while the mutant produced 66 milligrams per liter. We can see here from these results that there is a 330-fold increase of acromycin A production after deletion of the ACCJ gene. The results underscore the effectiveness of genetic manipulation for the improvement of fermentation titers. Antibacterial assays of the acromycin compounds compared with ampicillin antibiotic against gram-positive pathogens and clinical isolates showed that they exhibit good to excellent minimum inhibitory concentrations in the range of 1.5 to 12.5 micrograms per ml. Remarkably, the acromycins displayed superior activity compared with that of the ampicillin antibiotic against E. faecium K6039. Acromycins displayed MIC values of 3.1 to 6.3 microgram per ml, while ampicillin showed 25 microgram per ml MIC. Thus, the acromycins represent potential therapeutic lead molecules for the development of potent drugs against ampicillin-resistant Enterococcus clinical strains. Lastly, conclusions and future perspectives. Genetic manipulation will undoubtedly yield many more new and novel bioactive molecules from Streptomyces and might reboot the antibiotic pipeline. The acromycin pharmacophore offers promising potential for the development of acromycin-based antibiotics that target multi-drug resistant Enterococcus clinical isolates. And new drug leads with mechanisms of action different from those in clinical use are urgently needed to combat antimicrobial resistance. I would like to take this opportunity to give thanks to UP for the FRAS-DP program and the University of Aberdeen in Scotland, UK, where I finished my PhD. And to my supervisors, Dr. Haideng and Professor Marcel Jaspars, and to all my friends, thank you. And to everyone, thank you for listening. This ends my presentation.
Thank you so much, Dr. Maglangit, for the presentation on your research. No? And we also have invited somebody to react on this. He is a research coordinator of the College of Pharmacy from Adamson University. He is also a lecturer of natural products and medicinal chemistry on both undergraduate and graduate students. He obtained his BS in Pharmacy from the University of Santo Tomas, a master's in chemistry from the Ateneo de Manila University, and PhD chemistry from the University of Aberdeen. Let us listen to the reaction of Dr. Kevin Jace Miranda.
A pleasant morning to everyone. I am Dr. Kevin Jace A. Miranda, a faculty member at the College of Pharmacy in the Graduate School of Adamson University and the undergraduate research coordinator of the College of Pharmacy. I am here to give an insight on the presentation of Dr. Fleur-de-Lis F. Maglangit of the University of the Philippines, Cebu, on Streptomyces species MA37 and how this organism can help us to combat antimicrobial resistance. The presentation of Dr. Maglangit showed to us the power of genome mining and genetic manipulation on the discovery and expression of bioactive natural products, and how a single organism can be a prolific source of new and novel compounds. These new and novel compounds can combat antimicrobial resistance that we are facing right now. Streptomyces species MA37 was isolated from an unusual environment in Ghana and one of the few organisms that is capable of producing fluorometabolites, a phenomenon that is limited in nature. We have seen how this organism was able to produce new polypeptide antibiotics, the acromycins, and their bioactivity against pathogens of concern, particularly those that belong to the escape pathogens that were recognized by the World Health Organization as a growing resistance in all of our antibiotics available. Through genome mining, biosynthesis of the compounds was elucidated, and through genetic manipulation, the production of higher yield of a compound was possible. Dr. Maglangit's study showed to us the promising discoveries of novel bioactive compounds from extremotolerant environment, giving us insights on how these organisms adapt to this kind of environment. The future of natural products research to combat antimicrobial resistance lies on the uniqueness of the environment where these microorganisms thrive, producing different new and novel secondary metabolites. The Philippines, a nation where in the center of biodiversity is found, is really blessed with different species of microorganisms that can be utilized for natural products and drug discovery. This is to address the growing antimicrobial resistance cases that we are experiencing right now, particularly in this time of global public health crisis. Thank you very much.
Thank you so much, Dr. Kevin J. Miranda from Adamson University for that reaction. And that ends the researches and the presentations from our faculty members of the Department of Biology and Environmental Sciences. So let's give them a virtual round of applause for a tedious job well done. No? It's not an easy feat to research, but they have succeeded. And now we move on to our next set of speakers from the Department of Mathematics. Before we continue, may I remind those who are part of this Zoom meet to please drop your questions, queries, clarifications, comments, or any reactions on the researches presented on our chat box so that we can read them afterwards, as we have allocated a few minutes for an open forum. Our next speaker is an assistant professor of mathematics at UP Cebu. He finished his PhD in mathematics in 2020 at the Mindanao State University Iligan Institute of Technology with a scholarship under the Department of Science and Technology in the Accelerated Science and Technology Human Resource Development Program. His research is on algebraic hyperstructures. And let us listen this afternoon to Dr. Ramesses Manzano.
Good afternoon, everyone. First of all, I would like to thank the DOST as well as the University of the Philippines Cebu for this opportunity to present in this virtual conference in line with the National Science and Technology Week celebration. My dissertation, by the way, is entitled on pseudo-hypergear algebras, which I worked under the supervision of my dissertation advisor, Dr. Gaudencio Pitalcorin Jr. of MSU Iligan Institute of Technology. So to begin with, I will give you an overview of what is my paper all about. This paper introduces an algebraic structure, which we call pseudo-hypergear algebra. So this hyperstructure contains two hyperoperations. And moreover, different types of ideals is mentioned also here, some homomorphic properties, the quotient structure, the hyperproduct, and some isomorphism theorems are proven. Okay, so my study seeks to introduce a new class of hyperalgebra, which we call the pseudo-hypergear algebra. So the following are the objectives of the study: to introduce the concept of pseudo-hypergear algebras, to establish properties of this hyperalgebra, to construct the quotient structure, to provide results on hyperproducts, and to prove some isomorphism theorems. Okay, so this would be my working definition. We define a pseudo-hypergear algebra to be a non-empty set H equipped with two hyperoperations and a constant zero, satisfying the following axioms. So there are actually 5, for which X has a hyperorder with Y if and only if 0 is in X dot Y and 0 is in X star Y. And for every subsets AB of H, A has a hyperorder with B, means that for every A in A, there exists a B in A such that A has a hyperorder with. So in this Cayley table, coming from H containing 0, 1 to 3, So this is an example of a pseudo algebra, and that can be verified by routine calculation. So another example is the infinite set, the natural numbers together with zero, defined with these two hyper operations. So this is again a pseudo hyper-GR algebra. So the nice thing here is when the two operations coincide, then the pseudo hyper-GR algebra would turn out to be a hyper-GR algebra. Another class of a pseudo hyper-GR algebra focus on my research is all about the pseudo-star faithful, which is having this property: if 0 is an A star B, this implies that A has hyperorder with B. So this is an example of a pseudo hyper-GR algebra that is also a pseudo-star faithful. All right, so we define first these two sets, as we will be using this in defining ideals. Right. So let I be an empty set. It is called a pseudo hyper-GR ideal if those two sets is a subset of I. So for example, in the previous pseudo hyper-GR algebra, in the previous slides, if you let I to be the set containing 0, 2, so we can verify that it is a pseudo hyper-GR ideal. And the definition there, so ideal is actually the ideal of type 1 because there are 11 more. So this short list is a list of all pseudo hyper-GR ideals of each. And the following theorem gives the relationship among these ideals. So every ideal of type 2 is of type 1, type 4 is of types 1, 2, and 8. Every type 8 is of type 5, 6, 7, and 12. Every type 6 is of type 5 and 10. 7 is of types 5 and 11. 5 is of type 9. Type 12 is of types 9, 10, and 11. Type 10 is of type 9. Type 11 is of type 9. And this diagram shows the relationship among the pseudo hyper-GR ideals of H. Now we will see here how the quotient structure is created or constructed. This is actually constructed by a congruence relation. So if theta is a congruence relation on a pseudo hyper-GR algebra H together with I with the equivalence class of 0 under theta, we define H mod I to be the set of all I sub x, where x is in it. This is actually the equivalence classes of the set where your I sub x is equal to the equivalence class of x under theta for all x. And we define the hyper operation as follows, together with the hyper order. So the set H mod I together with these two hyper operation, and I is the quotient pseudo hyper GR algebra. So this is the homomorphism theorem that will lead us to the first isomorphism theorem. It states that if theta is a regular congruence relation on H and I will be the equivalence class of 0 under theta, this map is a homomorphism such that the I is equal to the kernel of F, then we have the conclusion H mod the kernel of F is isomorphic to the image of F. And if you have a pseudo hyper-GR algebra K of H with a regular congruence theta on H and I with the equivalence of 0 under theta, then if we define K mod I to be that set, then K mod I is a pseudo hyper sub-GR algebra of H mod I. So this corollary will lead us to the third isomorphism theorem. If you have two congruence relations with J, and I be the congruence class of 0 under theta 1 and theta 2, then J mod I is a pseudo hyper sub-GR algebra of H mod I. All right, so we have this lemma. Also, defined a theta on H mod I by I sub x is theta related to y if and only if your x is theta related to y. All the set in H mode I, then theta is a regular congruence relation, and the equivalence class of I under theta is actually your J mode I. So this is the third isomorphism theorem. Suppose you have two regular congruences on H with J and I, the congruence class of 0 under theta 1 and theta respectively, then H mode I, mode J mode I is isomorphic to the to H mode J, where J mode I is the set of I sub x in H mode I such that your x is in. So we define now the hyperproduct of two pseudo-hyper-GR algebras to be, we define the hyperoperation from H1 cross H2 cross H1 cross H2 going to the non-empty subsets of H1 cross H2 by this hyperoperations. Okay, so this is done component-wise as well as the hyperorder, which is also executed component-wise. We call the H1 cross H2 together with this two hyperoperation and this pair of zeros. This is called the hyperproduct H1 and H2. And the nice thing here is the hyperproduct of the pseudo-hyper-GR algebras H1 and H2 is also a pseudo-hyper-GR algebra. That ends my presentation. Thank you so much for listening. Have a nice day.
Thank you so much to Dr. Manzano. That was so a lot to take in. And we also have invited somebody to react on his research. He is a faculty member of the Cebu Normal University. He finished his PhD in mathematics at the Mindanao State University Iligan Institute of Technology with a scholarship also under the Department of Science and Technology. He published several articles and research papers at various national and international conferences in mathematics. His dissertation is entitled Forcing Subsets for Some Types of Sets in Graphs. Let us now listen to the reaction of Dr. Chris Armada.
Good day, everyone. This is a reaction in the paper entitled Pseudo-Hyper-GR Algebras, presented by Dr. Rameses Manzano Jr. The research pertains to the study of an algebraic structure. The study theorizes and sets out to further analyze this specific notion under the field of abstract algebra. This specific study is a great addition to the field of research as its ideas are certainly aligned with the continuing development of algebras. In contrast to other researches, this study is entirely independent from and completely uninfluenced with today's pandemic situation. As for most, this requires focus and the rational minds of the authors to derive and present accurate information and obtain significant results. Hyper-GR algebra was first defined and studied by Dr. Gaudencio Pitalcarin Jr. and Dr. Raymond Indaman. GR algebra may stand for Gaudencio-Raymond algebra, but was not mentioned in their paper. This observation is based solely on my perusal of the paper and its contents. Hyper-GR algebra has only one hyperoperation. On the other hand, Dr. Rameses Manzano Jr. and his advisor, Dr. Gaudencio Pitalcarin Jr., defined and studied the pseudo-hyper-GR algebra, which involves two hyperoperations and a set of actions that come in pairs or a combination of both hyperoperations, making it more interesting than the other established algebras. There is a study that the algebraic structures can be a useful mathematical tool both for the study of social relations from a static point of view and for the study of the social dynamics leading to the formation of coalitions. There are some applications of algebraic hyperstructures for the management of teaching and relationships in schools. Hence, for the next interested researchers, it is suggested to look for the applications of this concept, the pseudo-hyper-GR algebra, to some suitable real-world situations or problems. I would like to extend my appreciation to University of the Philippines, Cebu, for this opportunity. I am Dr. Crystal Armada from Cebu Normal University, and it is a great privilege for me to put forward my reaction to Dr. Manzano's paper entitled Pseudo-Hyper-GR Algebras.
Thank you so much, Dr. Chris Armada from CNU, which is also a state university based here in Cebu. Thank you so much for those who dropped their check-ins in the chat box. They are saying where they are from and while watching this webinar. So stay tuned until the end. And we have a photo opportunity also and a short Q&A if there are any questions that will be posted or typed here in our chat box. Now, our next speaker is an assistant professor in the Mathematics and Statistics program of the University of the Philippines, Cebu. She obtained her Doctor of Philosophy degree at the Institute of Mathematics, UP Diliman, last July 2021, while she finished her Master of Science in Mathematics and Bachelor of Science in Mathematics, both from the University of San Carlos, Cebu City. She was also a DOST scholar in both her PhD and BS degrees. Her research interests include mathematical models focused on population dynamics and infectious diseases in random networks. Ladies and gentlemen, let us now listen to our next speaker, Dr. Sherilyn Alota.
A pleasant day to all. I am Sherilyn Alota, and it is my pleasure to share with you my dissertation study in collaboration with my advisor, Dr. Carlin Arceo from the Institute of Mathematics, UP Diliman. Infectious diseases continue to threaten the lives of the human population. The spread of infectious diseases such as measles, influenza, HIV, and even the recent COVID-19 varies depending on the direct or indirect contacts from one individual or vector to another. Disease transmissions and its epidemics have been widely and intensively studied, especially when a new virus emerges as a cause of an increasing number of deaths in a population. Mathematical models of epidemics have been developed since Bernoulli's model on smallpox in 1760 and the classical SIR model of Kermack-McKendrick that assumes a homogeneously mixed population, creating compartments of individuals. It is very common for infectious diseases to have a latent period where an individual is infected and they remain asymptomatic and unable to transmit infection to susceptible individuals. Individuals under this time period are classified under an exposed class. In our study, we consider an SEIR model in a closed population and a disease transmission through direct contacts. We further assume heterogeneity in the population in terms of the number of contacts through which we use network or edge-based methods in describing and analyzing the disease transmissions, taking into consideration a static network where the edges are randomly formed by a given probability and are fixed as time increases. Other assumptions we consider in this study are as follows. Consider a population of fixed size n which will approach to infinity, and it is partitioned into 4 exclusive states, namely the susceptible, exposed, infectious, and recovered individuals. Due to immunity to being reinfected, recovered individuals cannot infect their neighbors. We use the proportion of each state in the derivation of the equations to associate it with large graph limits. Adding each of the normalized population yields a total of 1. We describe the population as a static random network with the individuals represented as nodes and the contacts between them as edges. A Molloy-Reed's configuration model is used to generate the aforementioned network, of which we specifically use a Poisson or a scale-free network, disregarding self-loops and multiple edges. The model dynamics of a disease propagation and an SEIR model considers the following assumptions. An infectious node transmits infection to each of its susceptible neighbors independently at a constant rate R. Exposed nodes become infectious at a constant rate alpha. Once infectious, a node recovers and becomes immune at a constant rate beta. A susceptible node cannot be infected by someone who is incubating, and this means that a susceptible node can only be infected by infectious nodes. Techniques presented by Volz in one of his papers are used in the derivation of the SEIR ODE model. In particular, the probability generating functions are utilized to express network values like degree distributions and its moments. These functions preserve the relationship between the nodes and edges in the network. In finding the basic reproduction number, we use the next generation matrix method. As for the simulation results, we implemented MATLAB codes for the Poisson and scale-free network generation for the stochastic SEIR simulations using Gillespie's algorithm and for the numerical SEIR predictions. Many interesting real-life interactions or structures are described by very large networks. Studying this network structure with the assumption that the number of nodes in the network approaches to infinity, one may look at properties of random and finite networks. We then consider if a sequence of these growing networks converges and if a limit of such sequence can possibly be described. Thus, we introduce large graph limits to study the stochasticity of the SEIR dynamics and establish its convergence to a deterministic limit applying measure theory in the probability of random events and the notion of weak convergence involving tightness and uniqueness arguments to investigate the convergence of a continuous time stochastic SEIR model. In the process, the degree distributions associated with the SEIR evolution are described as finite counting measures. The stochastic behavior are described by stochastic differential equations driven by Poisson point measures, as well as martingale and semi-martingale properties. These are key factors we need as an initial step in examining stochastic convergence to a deterministic solution. The Markovian evolution of the dynamics is described by infinitesimal generators on the set of real bounded functions. We have here the SDE model that describes the evolution of the epidemic through the degree distribution from which we prove the existence and strong uniqueness of its solution in the Skorood space of Cadlock functions. This is the limiting value of each distribution process and is a continuous measure-valid process that satisfies the following system of deterministic measure-valued equations. As a consequence to the limiting process, we arrived at this system of SEIR ODE model with the following initial conditions. And this model is illustrated by the following node-based and edge-based flow diagrams. We also computed for the basic reproduction number as follows, as well as the final epidemic size. The SEIR ODE model is verified by comparing its predictions with the stochastic simulations of the SEIR dynamics in large networks. For all the simulations done in this study, we set the following numerical or parameter values. We started with a proportion of initial infectants, which considers the following cases. We did 100 runs of stochastic simulations and compared the ensemble average of these simulations with the unique solution of the ODE model. Corresponding to the random networks generated, we got the following basic reproduction numbers and the respective final epidemic sizes. Here are some of the simulation results that we obtained. We have here the comparison of the predictions using the ODE model with the stochastic simulations of the SEIR dynamics using a Poisson network under case 1. When considering the scale-free network type, we can see that from this figure that the agreement between simulations and the ODE is not as close as that of the Poisson network. A comparison of the corresponding total fraction of infections of the SEIR ODE models in both Poisson and scale-free networks is depicted in this figure. From the 3 cases presented in the previous slide, where we varied initial infecteds, we can see from this figure that the presence of exposed nodes at the start of the epidemic results in a delay in the spread of the disease. We obtained similar behavior in a scale-free network. To further observe how and which of the disease parameters greatly affects proportion of population over time, we vary the disease parameters with the following conditions shown in the figure. Varying the exposed rate alpha with the following values, we observe that the smaller exposed rate gives a longer latency period, which leads to slower growth of the initial outbreak. Increasing alpha leads to a decrease in the latent period, and this results in a decrease of the peak of fraction of exposed nodes and eventually becomes almost zero. This limiting behavior where alpha tends towards infinity leads to the convergence of the SEIR dynamics to the SIR case. In conclusion, in the study of infectious diseases, one considers realistic factors to model dynamics of the disease transmission and its control or prevention. Specifically, we consider an exposed population in our model and integrate networks in disease transmission that allows us to describe the heterogeneity of the SEIR model, focusing more on the edges, investigating also the stochasticity of the spread of infectious diseases helps us to better understand its convergence and derivation of the deterministic or large graph limit. In this way, we can have a good approximation of the SEIR disease dynamics. The accuracy of the SEIR models we derived and their predictions guides us in extending this study, incorporating control interventions and prevention strategies of infectious diseases with latent periods. Moreover, the stochastic SEIR model and its convergence to a large graph limit emphasizing the network connectivity strives to understand its epidemiological features that may be extended and applied to particular infectious diseases such as COVID-19 or its variant. Additional factors like specific population compartments and control interventions, variability of the disease parameters, and network methods may be incorporated in the model to further analyze the spread of the disease. Here are some of our main references, and I would like to thank DOST, UP OVPAA, and UP Cebu for all the support in helping me finish my PhD degree, as well as in the dissemination of the results of this study. Thank you for listening.
Thank you also, Dr. Alota, for that extensive research. And we also have invited someone to react on this. She is a graduate of Applied Mathematics at the University of the Philippines, Mindanao, based in Davao, where she graduated cum laude and was also awarded as Best Special Problem and the class valedictorian in 2007. In 2011, she was awarded a Master of Science degree in Applied Mathematics at the University of Washington, based in Seattle, USA, and as a fellow under the Ford Foundation International Fellowship Program. And in 2017, she obtained her PhD in Interdisciplinary Studies, specializing in mathematical biology at the University of British Columbia in Canada. At present, she is serving as a tenured full professor of applied mathematics and chairman in the Department of Mathematics, Physics, and Computer Science at the University of the Philippines, Mindanao. Let us welcome and listen to her reaction, Dr. May Ann E. Mata.
Hi, everyone. The presentation of Dr. Alota is an example that demonstrates how a stochastic model would converge to a deterministic model in the context of systems that can be represented using graphs. And this is where I am going to focus my reaction on her work. So the phenomenon where stochastic models converge to deterministic models at large size limit is actually not new. It has been observed even in disease models represented as differential equations. Consider, for instance, the stochastic version of a susceptible infected recovered or SIR model with demography whose diagram is shown on the current slide. Based on this diagram, one can write the stochastic rates associated to all possible states and then formulate the corresponding stochastic model as a continuous time Markov process. For example, the corresponding probability of the event where S transitions to S plus 1 is given by the rate mu times n multiplied by the time step delta t plus some function of delta t. And we similarly define all these probabilities for each transition of events. It turns out that we can utilize these probabilities to yield stochastic equations that is comparable to the deterministic model. According to Bailey in 1975, we can find that each increment of the stochastic process can actually be decomposed into the sum of the expected value of the increment and the sum of centered increments. And so we write the following equations for the increments of S and I, which are our stochastic processes, in terms of their expected value and Poisson increments, because delta S and delta I are count processes following Poisson distribution. Theoretically, these conditionally centered Poisson variables may be replaced by what we call Brownian motion as rescaling is introduced. This is because for small delta t and large n, the process with centered Poisson increments is well approximated by the Gaussian increments having the same conditional variances. This technique gives rise to the stochastic differential equation formulation of the model. And it is not hard to see that from the stochastic model, if we let n go to infinity, we will arrive at the deterministic version of the SIR model with demography. In relation to Dr. Alota's presentation, her findings that the stochastic simulation of the large Poisson network representation of the SEIR model best verifies the metrics obtained in the deterministic version of SEIR model is not surprising at all, for a fact that intuitively, such a phenomenon has already existed before. But what is surprising to me is the manner of how she arrived at her findings of approximating an SEIR model with large graph limit, wherein she combined sophisticated mathematical approaches to arrive at her conclusion. I actually applaud her work for its novelty, as it paves way to open problems such as the question and the expected results for a large graph limit of disease network models with control interventions, which is actually or practically useful for studying diseases like the current pandemic that we are in. So that's all I have to say. Thank you very much for this opportunity, and may you all have a great day.
Thank you so much, Dr. Manta, for that equally comprehensive reaction to Dr. Alota's research. And should you have any questions, clarifications, or queries on the research, you may type them now if there are any. There is one comment from Jeremy Castro, who said hello to Ma'am Cherry Alota and sends her gratitude. Thank you for sharing your research. It is also very practical and applicable, especially in this time of the pandemic. And there any more questions, clarifications, queries, and comments? If there are none, our Teaching and Learning Resource Center has prepared an evaluation link for us who attended this event this afternoon, so that you may also receive your certificate. Let me just read their reminders. The evaluation and processing of e-certificates are automated, so make sure you type in the correct spelling of your name since this will reflect on your e-certificates. And also, please be mindful in typing your email address because this is where your certificates will be sent. We suggest that you keep a copy of your response in the evaluation so that we can verify if there are any mistakes made. Normally, it only takes 3 minutes to fill up the evaluation, and after submitting, you will receive your certificate. Should you not be able to receive it by 5 p.m. today, please send a reply message to the email and which is msgalorio@up.edu.ph, and the team from PLRC will manually send the certificate the next working day. So it should be very easy. And also another reminder, the link for the evaluation is open for only 15 minutes. And once the time is up, they will close the evaluation link, which is now posted on the group chat. So by this moment, you can access it and answer it for the next 15 minutes to receive your certificates. The researches presented were all very dynamic and informative and relevant in today's time, tackling on environmental issues such as forest fires, the problems in our marine biodiversity, and etc. Also, it has been proven that it can be very applicable to addressing unprecedented events such as the pandemic. And that's why we have to keep on and carry on, keep on dreaming, keep on researching, because like what was mentioned this morning, without science, there will be no hope. While there is science, there is still hope, and we continue to support. And UP Cebu is also very open to future partnerships, collaborations, to any of the researches that was presented and will be also done in the future. You may get in touch with our different offices with regards to the researches. You may search our socials on Facebook and our UP website. Please stay at the end of the program because we are to conduct a very short photo opportunity since it is not normal for us to be in one Zoom room with equally competent researchers and PhD candidates. All right. At this moment, as UP faculty and students, if there are any, we all know this song by heart. Let us all sing the UP hymn, UP Naming Mahal. UP Naming Mahal, naman pa sa hirap. Mabuhay ang pag-asa ng bayan, mabuhay ang agham at teknolohiya, mabuhay ang UP Cebu. From all of us here at UP Cebu under the leadership of Chancellor Lisa Coro, we want to thank you all for attending this day's activities for the National Science Technology. We invite you to a photo opportunity, and I'm turning over the virtual floor to our public information officer.
Hi everyone, kindly turn on your video so that we can have a small photo up. We have 3 pages, so we don't know what page we'll be landing on, so let's all just smile. Waiting for page 1 to fill up. Okay, sige, everybody, smile for page 1. Smile, everybody. 1, 2. Okay, page 2. Smile. Then page 3. 1, 2, 3. Salamat sa tanan.
Thank you so much again, everyone, for attending today, this morning, and this afternoon. There's still day 2 tomorrow, so I hope you can also join us as we continue to celebrate the National Science and Technology Week. This has been Annie Perez Gallardo from the College of Communication, Art, and Design saying good afternoon and stay safe. It's raining outside from my area here in Consolacion, so I hope you keep dry and stay safe. You may log out of the Zoom room once you have finished your evaluation. Thank you.